Ladies and gentlemen, please join our hands together to welcome the laureate of the 2018 Tang Prize in Biopharmaceutical Science, Dr. Brian Druker from Oregon Health Science University, accompanied by Chancellor Wenhua Li of China Medical University, Vice President Lu Haiwang of China Medical University, Superintendent Anli Zheng of National Taiwan University Cancer Center, Vice President Hong Ming-Qi of the University of Texas MD Anderson Cancer Center, and CEO Zheng Chuan-Chen of the Tang Prize Foundation. Welcome. Good afternoon, distinguished speaker, honored guests, and participants. Welcome to the Masters Forum of the 2018 Tang Prize in Biopharmaceutical Science. I'm Roting Lin from China Medical University. It's my honor to be the MC of this forum. On behalf of the China Medical University and the Tang Prize Foundation, I would like to extend a very warm welcome to all of you. To begin with our program, we are pleased to have Chancellor Wenhua Li of China Medical University to deliver the opening remarks. Please join me in welcoming Chancellor Wenhua Li of China Medical University.
Distinguished guests, ladies and gentlemen, welcome to the CMU. And it is really a distinct pleasure to have this great chance to host a Tang Prize Forum. And particularly that we have this year's laureate, Dr. Brian Druker, and who is the bona fide medical doctor, okay, and hematology oncologist, okay, that related to the CMU closely come to deliver this keynote speech. And not only that, we also have the Professor Anli Zheng, academician Ming-Qi Hong, and academician Lu Haiwang, okay, come with us to deliver today's panel, and then for everybody to ask questions. And we are particularly grateful to the Tang Prize Committee, okay, for them to give us this opportunity, okay, to host this special forum. And you know, this year is CMU's 60th anniversary, and that's exactly fit to our celebration schedule, and that as a chancellor, we really appreciate this great opportunity. And I hope that today we have allowed the medical students and allowed the PhD students and also allowed the faculties and come all over the place and to enjoy today's forum. I hope you will enjoy this symposium as much as you can, because this only comes once in your lifetime. Thank you very much. Thank you.
Thank you, Chancellor Li, for the wonderful remark. I'd like to invite Chancellor Li to introduce our esteemed speaker.
I'd like to introduce you laureate, Tang Prize laureate, Dr. Brian Druker. Brian graduated from UCSD, bachelor and medical degree. And later, he went to St. Louis, WashU, for his clinical training. And after finishing his clinical training, he went to the Mecca of United States. That's in Boston area. He went to Harvard, work with Tom Roberts and Jim Griffin. And at that time, he really received a lot of training in terms of basic science. and particularly carcinogenesis. And he has published many, many papers during that time. But he did not stay at Harvard. He went to Oregon State University, and right now it's Oregon Health and Science University, and to serve as a faculty in 1993. And the important discovery is exactly he has done in the Oregon University. Okay, and he was quite young, okay, around the age of 40s around, and he make a historical discovery, okay, around 2000. He report the first time, okay, in the history that today he is talking about the immutable as the paradigm of the target therapy that use this drug to treat the chronic myelogenous leukemia, CML patients. Originally, this disease essentially is hopeless, but he able to extend the first patient, and at a time when I visited him, it's already 21 patients that can sustain for many, many years. This kind of historical breakthrough make everybody eye-opening. And indeed, those are extremely important work. And because of those important work, he received many, many recognitions. He received, such as a GM award, that's the cancer research is a really quite good award. And later, the Lasker Dubegi Award, and then Japan Prize. Now, this year is 10 Prize. Of course, there's another one, it's called the Nobel Prize, pretty soon, I guess. Okay, so this illustrator, a physician devoted in the basic science, not only can help patients, but also really can make a breakthrough, okay, in terms of medical history. And that, I think for us, there are many medical students here and many PhD students here, then you understand this kind of discovery, as soon as you are devoted like Brown, you have chance. Okay, and with this great pleasure, and I really pleased and a great honor, okay, to have the Brown to deliver this talk. Thank you.
Thank you, Chancellor Lee. It's now our great pleasure to invite Dr. Druker to present us imatinib as a paradigm of targeted cancer therapies. Welcome, Dr. Druker.
Thank you very much. It's a privilege for me to be here. I want to thank the Tang Foundation for this recognition. I want to thank Chancellor Lee for inviting me to CMU and getting to see the wonderful university and the city here. What I want to do in my talk today is what Chancellor Lee asked me to do, which is to try to inspire you. So to do that, I want to go back a little bit. And you might think that this, as you've heard, this is not the first award that I've received. So I have to take you back a little bit in time. And this was my very first award for science. And yes, please go ahead and laugh. Look at the white shoes and the high water pants and the big ears. Okay, I own it. I was in middle school. I was in 6th grade and I won my science fair. And I'm proud of that. But what I want you to recognize as you think about your own careers is if they take nothing else away from my lecture today, I want to try to give you the courage to ask big questions. Big questions that can change the way that science is done, the way that patients are treated. So we'll start with a big question. What? What should I work on? What are the big problems? What I decided to work on was a disease called chronic myeloid leukemia. So what is chronic myeloid leukemia? Well, we have to go back to 1845 when two pathologists, Virchow on the right and Bennett on the left, who described the first 2 cases of chronic myeloid leukemia. Now this is 1845, and thereafter there ensued a debate about who was really first. So nothing new in science. We still debate about who was first, who made the first discovery. They did it back in 1845, and ultimately Virchow said in a public lecture that Bennett had described a case of leukemia 6 weeks prior to his, so Bennett gets the credit for being first. So what is chronic myeloid leukemia? It's a relatively common leukemia, making up about 15 to 20% of all leukemias. There are about 1,000 to 2,000 cases per 100,000 people per year, and the incidence is pretty level worldwide. So the number of cases in Taiwan are about the same number incidence in the United States. The average age of onset is about 50 years of age, although it can affect any age group. Historically, and I'll just have you remember this, people used to live about 3 to 5 years. So if you were diagnosed with this leukemia, your doctor told you, we expect you have 3 to 5 years to live. So the next big question, why? And I want to take you through some historical examples of people that had the courage and insight, just have curiosity about what was around them in the world. And we all are surrounded by mysteries. We're all surrounded by commonplace things. But sometimes it's just a matter of asking a big question like, why? So let me start with a non-medical example. Velcro. And let me tell you about the origins of Velcro. I'm showing you George de Mestral, who discovered or invented Velcro. And one day he went hiking in the woods in the Swiss Alps. He was wearing a velvet vest, and on the way back from his hike when he got home, he saw that his vest was covered with burrs. And he did what millions, if not billions, of people have done throughout history that have gone on hikes and gotten burrs on their clothes. He picked the burr off, and he probably pricked his finger, and it probably hurt and drew some blood, and he swore at the thing. But he asked a question. Why does this burr stick so tightly to my velvet vest? Why? Turns out he's an engineer. He went down into his basement. He had a microscope, and he looked under the microscope. And his velvet vest had loops, and the burr had hooks. And he said, I bet I could make a fastener out of that. That turned into Velcro. I'll give you a medical example. Penicillin. Alexander Fleming walked into the lab one day. He was growing some bacteria on a petri dish, and he noticed that there was yeast contamination. Instead of throwing out that petri dish like millions of researchers have done throughout history, he looked at it carefully and noticed that the bacteria weren't growing very well around the contaminated yeast. And he asked why. Why aren't those bacteria growing? Now, it turns out he had spent much of his career, he was a surgeon, he had worked in World War I, and he grew tired of pulling bullets out of wounded soldiers only to have them die several weeks later of infection. So he dedicated his career to finding a better way to treat infections, and 12 years later, he walked into the lab, saw this contaminated plate, and asked why. So throughout history, people have looked at this abnormal blood smear from a patient with CML. And what you see are many, many white blood cells than you normally would see in the bloodstream of a patient, and many, many immature cells. So the question is, why does that happen? What fuels the growth of these white blood cells? Why are they growing abnormally? And throughout history, at least the recent history from the 1960s on, people have asked why this has happened. Two pathologists again, Peter Knoll and David Hungerford, working in Philadelphia, United States, looked under a microscope at the chromosomes in patients' blood and bone marrow with chronic myeloid leukemia and identified a shortened chromosome 22. And they published this landmark paper in Science in 1960. This is the entire paper, 3 paragraphs, 300 words. That's right, quality and quantity do not equate. But look at the last paragraph. They said the findings suggest a causal relationship between the chromosome abnormality observed and chronic granulocytic, which is what is called in those days now chronic myeloid leukemia. At the time in 1960, most people didn't believe that what they said. They figured that this was an associated abnormality, had nothing to do with the cause, but we have later learned that they were absolutely right and way ahead of their time. In 1973, Janet Rowley, working at the University of Chicago, asked again, why is this chromosome short, this chromosome 22? And what she discovered was it was short because the long arms of chromosomes 9 and 22 exchanged segments. So that all of this part of chromosome 9 was translocated to 22, made a short chromosome 22 and a long chromosome 9. Now in the 1970s, 1980s, oncogenes were being discovered, and people asked where? Where do these oncogenes live? On what chromosome? And what people realized is the Abel oncogene, which is located on chromosome 9, was translocated in its entirety to chromosome 22, forming this fusion gene and protein called BCR-ABL. But the reality is that if you look at the history of chronic myeloid leukemia, there have been thousands of physicians and scientists who've asked why and who've contributed to our knowledge of what drives the growth of this leukemia. We start with all of tumor virology, which led to the discovery of the Rous sarcoma virus, which led to VSARC and VABL. So viral Abelson, that's the Abel tyrosine kinase. That came right out of the discovery of oncogenes and the identification of BCR-ABL. I showed you the work of Peter Knoll, David Hungerford, and Janet Rowley, which is chromosome banding, gene mapping. It led to the identification of this chromosome translocation. There was also the entire field of protein phosphorylation. In the 1930s, we understood serine and threonine kinases. And in 1979, my colleague and fellow Tang Prize recipient, Tony Hunter, identified tyrosine kinases. As it turns out, the first tyrosine kinase he identified was VSARC. The second one was VABL. What that allowed us to do over the course of almost a century was to think about, could we shut down the activity of these tyrosine kinases? So to bring all this together, when I entered the field in about 1988 to 1990, we had BCR-ABL as a therapeutic target. It was the product of the Philadelphia chromosome. It was present in all patients with chronic myeloid leukemia. From animal models, we knew, that was actually George Daly, Rick Van Etten, and David Baltimore's lab, showed that if you put BCR-ABL into an animal, into a mouse, they got leukemia. One of the first experiments I did with Jim Griffin was to show that if you knocked out the kinase activity of this protein, it lost all of its transforming function. So this kinase activity was absolutely required for its activity. So the first question I ask when I started along this path is, how are we going to target this molecular abnormality? In the early 1990s, there were three possible ways. Actually, there were really only two possible ways. One of them was antisense oligonucleotides. In 1988, 1990, antisense oligonucleotides were going to cure every disease. You could get them to bind specifically. You could knock out targets and downregulate the expression. This was going to cure everything. But I looked at that and said, this is an engineering problem. You got to figure out how to deliver this to cells, how to regulate expression, how to keep expression high. And I thought, that's an engineer's problem. That's not a biologist's problem. The other thing that was absolutely the rage in the early 1990s was antibodies. We had just seen the first antibody therapies come along, and the view was we could make an antibody to this BCR-ABL junction peptide, but to me that was an immunologist problem, and I wasn't an immunologist. I had trained as a chemist and a biochemist, and so I thought, why not attack the enzymatic activity? Again, that was my background. The problem was that kinases were not a viable target in 1990. So despite the fact that there are 50 FDA-approved kinase inhibitors on the market today. In 1990, the general view was a kinase inhibitor would never be successful. And here's why. First of all, Tony Hunter had predicted that there'd be about 1,000 kinases in the kinome. And if you shut off one kinase with an ATP competitive inhibitor, the view was you'd inhibit every single one of them. Because ATP is ATP, how are you going to get something that's going to have specificity? So that was what chemical biologists said. Oncologists are a pretty skeptical group. We hadn't seen many cures in the 1980s, and the view was, when will single-agent therapy for cancer ever work? And biochemists' view was, you got micromolar concentrations of ATP in a cell, and you'll never have a competitive inhibitor. Third problem was there was concerns about toxicity. Even if you could get specificity, some of the early knockout animals, you took out one kinase, embryonic lethal. So the view was, even if you had specificity, this could be incredibly toxic. But the biggest hurdle, 5,000 patients a year in the United States, it's never going to make any money. So if you think about it, the drug companies say it costs $1 billion to develop a new drug, and you have a 1 in 10 chance of success. And if you look at the market size and you replace the current drug on the market, the view was this drug would make $100 million a year. So I doubt anybody here is going to invest in $1 billion in a project with a 1 in 10 chance of success, and you get $100 million a year with maybe 10 years of exclusivity. You'll never make back your investment. Well, I'll fast forward and tell you that the drug that we developed ultimately made $40 billion for the drug company Novartis. I don't see any royalties, and I'm okay with that. But the marketer people were absolutely wrong. Despite all these hurdles, in 1990, so just to come back to how this, my thinking about this is a tyrosine kinase. B-cerebral kinase binds ATP, transfers phosphate onto tyrosine residues, and that causes the uncontrolled proliferation. So my view is pretty simple. Just block binding to this kinase and you'd have an ideal therapeutic drug. In 1993, my colleague Nick Lydon, who's working at Novartis, sent me a handful of compounds, and our laboratory showed that this one, imatinib or Gleevec, was the absolute best at shutting down the proliferation of CML cells both in vitro and in vivo. And 20 years ago, 1998, we entered clinical trials. And with a 5-year follow-up, you can see we have a 95% 5-year survival. So remember, patients have been told you have 3 to 5 years to live. Now with a once-a-day pill, patients have a 95% 5-year survival. So let me show you what that means for somebody.
I'm very excited to be here tonight. I'm going to start out by telling you all a little bit about myself. I'm 18 years old. I'm a nursing student at University of Portland. I graduated in the top 8% of my high school class. Thank you. I was part of the Royal Crowns dance team where I was team captain, all-state athlete, and state champion. I love my dogs, Italian food, and Grey's Anatomy. And when I was 6 years old, I was diagnosed with chronic myeloid leukemia. We all know what happens in the treatment for cancer: chemo and radiation, hair loss and nausea. But would you like to know a secret? I never had any of that. I had Gleevec. Because of Brian Druker, my life is as I described it to you. Dr. Druker began developing Gleevec the same year that I was born, and it was FDA approved just 1 month before I was diagnosed. And if you want to talk about timing, that's some of the best that I've ever heard. It's because of the Knight Cancer Institute that this timing is possible. It's because of the Knight Cancer Institute that Dr. Druker was able to develop this groundbreaking new drug. It's because of Brian that I got to carry a first place trophy back to my dance team at state. And it's because of the Knight Cancer Institute that I'm going to school to become a nurse. Can you imagine? a world where cancer is merely a memory. Brian Druker, Phil Knight, and you together are making this a reality, and I personally thank you.
Thank you. Katie is now 17 years on treatment, is a nurse at our Children's Hospital in Portland, and still continue to do wonderful things. If you look at the 10-year survival now, there's some really interesting data. And if you look at the middle column called major molecular response, these are patients who have a very deep response where it can barely detect any BCR-ABL transcripts. If you look at their 10-year survival, nobody has died of their leukemia. Now, unfortunately, some of our patients have died of other causes, but if you achieve this deep molecular response, the likelihood of you dying of your leukemia at 10 years is virtually nil. And that is absolutely remarkable to see the durability and the effectiveness of a drug like this for cancer. But imatinib is not perfect. If you look at 5 years, about 17% of patients will have disease progression or relapse on therapy. What we've learned over the course of 15 to 17 years is that most of the relapses, about 60%, are due to mutations of the target. The kinase domain picks up mutations that no longer allow the drug to bind. What that has allowed this pharmaceutical industry to do is develop 4 new drugs that target these resistant mutations. So now there are 5 drugs on the market for this leukemia that can target all the common mutations that we see. So to summarize this, we have current standard of therapy with imatinib, which significantly prolongs disease duration. We have relapses mostly due to kinase domain mutations, novel inhibitors with significant activity that are actually a little bit more potent and are being moved into frontline therapy. And we've converted this disease to a manageable condition. And even more remarkably, about 10 to 20% of patients who have undetectable BCR-ABL transcripts have been able to discontinue therapy altogether. And so there's a small fraction of patients who are actually cured. So where else has imatinib worked? We knew when we profiled the drug that it inhibited ABL, the PDGF receptor, and KIT, tyrosine kinases. KIT is activated by mutation in gastrointestinal stromal tumors, and imatinib has significant activity in that disease. About 2 or 3% of patients with melanoma have KIT mutations, and imatinib has activity in that subset of patients. And then there are 2 relatively rare diseases that are driven by PDGF receptor activation, the hypereosinophilic syndrome and DFSP, and imatinib also has significant activity in those diseases. So what lessons have we learned? The first lesson for me is the target's really important. And to me, you can make this pretty simple. If you have a good target and a good drug, you can get good results. It really can be that simple. But the reality also is that you need both sides of this equation. There have been many examples in the not too distant past where not so good drugs have made good targets look bad. So you need both a good target and a good drug to get good results. So how do you translate this now to other cancers? So the first task in my mind is you've got to get the right targets. And the right targets are going to be these early molecular pathogenic events like BCR-ABL. You have to treat early in the course of the disease. Remember, chronic myeloid leukemia has a 3 to 5 year lead time before it becomes an acute leukemia, which is much more like an advanced metastatic cancer. So people have looked at precision medicine and all these targeted agents for advanced cancer, like melanoma and lung cancer, where responses only last 3 to 6 months and said, there's something wrong. What I've said is, don't forget you got to treat earlier before this becomes an advanced heterogeneous metastatic cancer. And of course, you've got to match the right patient with the right drug. If you have activated ABL, you need an ABL inhibitor, and so on and so forth. So that's great, but what are we doing now to make this occur faster? And if you think about the recent history From the discovery of the Philadelphia chromosome until imatinib's approval in 2001, that was a 40-year process. It took almost 25 years to go from the discovery of the Philadelphia chromosome to the identification of BCR-ABL, and then another 15 years to go through the drug discovery process for an overall 40-year length of time, and we have to do better. So what my laboratory is focused on is this target identification part, and I'll let the drug industry work on the drug discovery part. But I want to take you through what we've done to try to accelerate progress of target identification. And right now, if you look at the scientific research world, particularly cancer research, is divided into two camps. The first camp says we need to sequence everything. We just need whole genome, whole exome, RNA-seq, and we're going to solve all the problems in the world. But the problem with whole genome and whole exome is you pick up a lot of wheat with the chaff, meaning you see all these passenger mutations, and you don't know which ones are really driving the cancer and which ones are just random mutations. Another group of investigators say what you need are functional screens. You need to understand what pathways activated in functional screens using CRISPR, RNAi. We've used kinase inhibitors. The problem here is you often will get a target, but you don't know why it's activated. So my lab has taken an approach of why not combine both of these? Integrate the results, prioritize genomic data, understand mechanisms from functional screens, and use that to accelerate progress. Now, of course, because I'm working with leukemia, I can get blood samples and marrow, so I make it a little bit easier than my solid tumor colleagues, but nonetheless, we've taken this integrated approach. So the idea is we'll get a patient sample, and again, these are all primary samples coming directly from leukemia patients. Not cell line models, not animal models, these are patient samples. We will sequence them, identify some mutations. We will run them through our functional screens, both siRNA, CRISPR, inhibitor screens, and we'll integrate all that data, prioritize targets for validation, and then use that information to devise treatment strategies. So what we did with one example, and we have now many examples, is in a 9-month course, we took a patient sample, ran it through all of this, and then published this paper in the New England Journal showing that in a patient with this myeloproliferative syndrome called chronic neutrophilic leukemia, that there were mutations in what's known as the CSF3R receptor, historically known as GCSF receptor. So what we showed was in a group of patients with this disorder, after we identified the first mutation, this is the defining molecular pathogenetic feature of CNL, much like BCR-ABL. Interestingly, from our functional screens, we knew the largest set of mutations were sensitive to JAK inhibitors, and there's one already on the market, ruxolitinib. So we were able very rapidly to treat this patient with ruxolitinib and very rapidly saw blood counts return to normal. We are now at the end of a multi-center trial to actually take this drug from our laboratory to FDA approval, and that trial is actually looking quite good in this very small subset of patients. But again, it shows you how quickly you can learn. So what's our job for the 21st century in cancer research? In my view, we have to identify all the molecular pathogenic events in cancer. We have to get better at diagnosing cancer earlier through improved diagnostic and imaging technologies so we can treat early in the course of the disease. We clearly have to get faster at discovering and developing drugs. And ultimately, imagine if we can understand all of our genetic risks, then we can actually think about molecularly targeted prevention for cancer. So as I think about this, we have to take a very broad-based approach to cancer. We need far more specific therapies directed at critical targets. We need much better prevention and early diagnosis. And we also need to better understand how to modify the immune system. We've seen a remarkable breakthrough with the development of checkpoint inhibitors, and we need to do better at understanding who they work for and how to make them work for more patients. So in closing, I'd like to thank lots of people that have gone on this journey with me, all the people who have helped me out at OHSU and the Knight Cancer Institute, many of the study groups that put patients on our clinical trials, Novartis who funded the clinical trials, and my own funding agencies, which have been the Leukemia and Lymphoma Society, the National Institutes of Health, and Howard Hughes Medical Institute. But the people I want to thank most are the people who were brave enough to enroll in the early clinical trials who went on this journey with me. And these patients, many of them are still with me, many of them still doing the things that they enjoy. Things like this longest patient who's continuously been on Gleevec for 19.5 years, enrolled in the phase 1 clinical trial, doing what she enjoys, which is gardening. The first patient from Italy enrolled in our clinical trials, doing what he enjoys, which is dancing. This woman who was referred to me from hospice in 2000 is still alive, spending time with her grandchildren and now great-grandchildren. Lots of people traveling to Oregon for our clinical trial, and some doing truly remarkable things like this woman who was the first patient from Australia treated in our clinical trials. She returned to Australia and was selected as one of the torchbearers in the Sydney Olympics in 2000. But in closing, I'd like to leave you with one last big question. Oh, I'm sorry. Before I get there, the reality is these individuals end up being large groups of people. So we treated one patient at a time, we treat one disease at a time, and ultimately we can make a big impact. But in closing, I want to leave you with one last big question. And that big question is who? And my answer is, why not you? Thank you very much.
Thank you, Dr. Druker, for giving us this inspiring talk. It really means a lot to all of us. Now we are going to have a 70-minute panel discussion. Please join me in welcoming our panelists to the stage. Chancellor Wenhua Li of China Medical University, Vice President Lu Haiwang of China Medical University, Superintendent Anli Zheng of National Taiwan University Cancer Center, Vice President Ming-Chi Hong of the University of Texas MD Anderson Cancer Center.
Thank you for Brian's very inspiring talk. I think the audience, particularly our students and PhD students, the last slides you have to remember who is you. So it's you. So you will be the next Brian, okay? So that's the waiting for you to make that discovery. Okay, now I think we are opening for the discussion. And so the first one, I will ask Ming-Chi to.
Well, thank you for this opportunity, and also it certainly is a great honor to be here with Brian together. I know Brian's story for a long time, and although this is this is not the first time we met earlier, and I can share one story with you that I talked to Brian earlier. That is, I was in Houston. Many of you know that. And then I also frequently to give a lecture on cancer therapy or general cancer knowledge in the Chinese community in Houston, non-scientific community. And one time, because I was working on breast cancer, HER2/neu, and EGFR receptor, and it's also another type of receptor, tyrosine kinase. Then one time, after I finished the talk, then there are many people who ask questions, but there was one gentleman, it's about 40-ish, and with a high school girl with him. Obviously, I can tell that's his daughter. And they were waiting for here a long time. I thought they would have questions for scientific questions. But actually, they were waiting for, very patient, waiting for there for a long time. Many people have questions. And at the end, both of them come to say thank you to me. And I was totally, I don't know why they're saying it to you. I said, why you say that? Because that girl was cured by Gleevec, developed by Dr. Druker, the one she just mentioned. So I told them that, I'm talking about HER2/neu. It's also a receptor. It's also a tyrosine kinase, but I have nothing to. I never work on Gleevec. But what the father told me, which I think it makes sense too, that he said, well, I'm coming here just because they wish they could say thank you to Brian Druker directly. But since they don't have that opportunity, but then when they heard that we are doing something similar, because, well, maybe later on I can say that story. So you would like to say thank you to the people who are doing this type of work. So obviously, Brian is the person who is really converted the whole field from chemotherapy, the non-specific chemotherapy, to the target therapy. I think this Gleevec drug was really the turning point. And then also because of the impact that to make the society, the general society, to really appreciate those people who are working on developing new drug to help the patient to cure the cancer. So again, I'd like to echo that the last slide when Brian say, who can do it? Think about it. Many of you are students or are young physician here. Pick up what you like to do in the future and then go for it. And it could be you. It could be you. So don't, how to say that in Chinese? Maybe I just take one Chinese now. 天生我材必有用. But who would like to follow that Chinese point? Who want to volunteer? After 天生我材必有用, what's the next one? You guys must have known that. Somebody want to volunteer? The student, the one with the glasses. You. Yeah. After that, what is that? 天生我材必有用. After that, what was supposed to be? Okay, he said 千金散尽还复来 because you are Li Bai, right? But he's right. But I would like to encourage everybody to think about it. After 天生我材必有用, please also remember 志向必高方有成. Okay, I just want to share this with you. Okay, 志向必高方有成. So you can be someone who are the who. That you can be someone who, as long as you're committed and pick up the important area, you could be someone who make an impact for the society. 志向必高方有成. 所以你们在实验室的时候，不要老是想说不来实验听说现在博士班的学生这个都不来实验室做实验。So that's not right. Okay.
The PhD student does not work hard. He's complaining the PhD student does not work hard. Okay, next, Andy, you want to talk? Yeah.
Thank you so much, Brian, for this excellent, inspiring talk. And I think sitting in this room, there are many scientists, young scientists, and also young physicians. And I believe many of them probably have some great idea, as you had some 20 years ago. But sometimes it's so difficult to sell the idea to the sponsors. So my question is, have you ever had any frustration doing so? Or how did you cope with this? To convince that this is a great idea, even it's against the business model. How can you do that?
Well, yes, there's a longer story there, and the reality is that Chancellor Lee talked about me leaving Harvard. The actual story was I was asked to leave Harvard because they didn't believe that my work was going anywhere. And so for me, that was kind of a moment in time where you have to ask yourself, do you believe what somebody who may be smarter than you are is telling you your work isn't ever going to work, or do you believe in yourself? And for me, that was that moment where I had to decide, who do I believe in? And ultimately, it gave me the courage to believe in myself. I moved to Oregon. Then I had a goal, and the goal was to get a drug into clinical trials for patients with CML. And yes, I faced enormous hurdles with the drug company that felt they would never make enough money. But ultimately, I had patients in clinic who needed this. And they needed somebody to speak for them. They put their faith in me that I could offer something better than what they had to look forward to, which was certain death. So I became their champion, and I continued to be persistent in trying to persuade the company that they needed to get into clinical trials. And ultimately, I devised a strategy after 4 or 5 that failed. I figured out a way to get them to say yes and move into clinical trials. And within 6 months, the drug was working in everybody.
Well, that tells you. And
so I can share with Brian some of my personal experience, because I tried to convince another company, Bayer, to develop sonafenib for liver cancer. So that was, I think it's a story that I can share with everybody, that when you try to do that, the company people would say, what is liver cancer? Because it's rare in Western country. So the big pharma is not interested in that, because this is going to be probably a small market. In the whole Asia-Pacific region, only 8% of the total sale for any anti-cancer agent. So any big cancer in this part of the world may not be important for the big pharma. So it really spent a lot of time for us to convince them. So I asked them that if some of you key person might come to Taiwan, then I can meet you. I try to explain to you why liver cancer is so important here. And they refused. So after almost a year later, then somebody is going to travel to Hong Kong. And I said, if you are in Hong Kong, please come to Taipei. It's very short distance. And they refused. They don't like to hear that. So later on, I traveled to Hong Kong, and I spent a whole day trying to convince them. And finally, we got the drug. So that is something that I think all the young investigators should learn, that you have to be persistent enough. Otherwise, you are going nowhere.
thank you, Annie. And that tells us nothing's easy, okay? But nothing's that difficult. That correct, right?
That is correct.
Yeah. So as you have that, have your mind, you believe yourself, and you are, say, very confident that you should give the benefit of patients, then you should try hard to get it. Okay. Now, Lukai. Okay.
Right. A remarkable achievement like this must have 100 wonderful stories. You show us that with the lovely 6 years old and going to 18 years, beautiful women, dancing women, and like the happy gardener, the grandmother. But I like to add one more today. My very dear, close relative was diagnosed CML 5-6 years ago, and I can tell you, she is still active and healthy today, and I thank you for her for that.
Thank you very much.
I'd like to turn to the 17% less fortunate one who developed imatinib resistance and then progressed to the breast, the accelerated and breast phase. Now, I'd like to address 2 aspects and then have your input. And this is not only for my interest, also for my relatives' interest. And that is a stem cell. How Gluvac is able to deal with stem cell. And the second aspect is not a mutation of the kinase domain, but activation for alternative pathway that make a cell become BCL-able independent. How do you deal with those cases?
I'll start with thank you for the question. So I'll start with the second. We know that 60% of resistance are due to mutations, and 40% are alternate signaling pathway activation. And our studies have shown that it's many of the same mutations you'll see in other leukemias. So RAS mutations are very common. Other transcription factors that we see, MLL, and other driving mutations you'll see in other leukemias are what we see as BCR-ABL independent mechanisms of resistance. But it's extremely heterogeneous in that 40%. The good news, however, beneath this is that I rarely see anybody actually progress to blast crisis. If somebody becomes resistant, usually we can transition to an alternate kinase inhibitor. And some of these kinase inhibitors aren't quite as specific as imatinib. So they also shut down some of the alternate pathways and have been quite effective at preventing people from progressing to blast crisis. But none of the drugs eradicate stem cells. And many, many years ago, our lab and many others have shown that you can shut down BCR-ABL activity in CML stem cells, and the cells essentially revert to normal. They survive. They rest and they're quiescent, but they're not killed. If they proliferate, they're dead. But if they're resting and quiescent, they're resistant to being killed despite the fact that the enzyme BCR-ABL is shut down. So we don't have a good way to eradicate those stem cells, and that's why people remain on therapy.
Thank you.
Okay. Is there any questions from the audience? Okay, you do. All right. Otherwise, I can ask a question to Brian for you. Okay.
Hi, Brian.
Right.
Nice story. So my question is, when you work on this tyrosine kinase inhibitor, I guess other people are also working on the same topic. And how can you pick up the right drug and then go through it? So can you share with us your experience?
Right. Well, so the reality is I didn't develop this drug. This drug was developed at a drug company. And that's what they're really good at. As I said in an interview yesterday, there was a famous American robber by the name of Willie Sutton. And when Willie was asked, why do you rob banks? He said, well, that's where the money is. So why do we work with drug companies? They're really good at making drugs. So they can go, if you think about the difference between a compound, which is something that might inhibit a kinase, versus a drug which you can give to people, it has to have a good half-life. It has to be able to be absorbed. It has to be stable. It has to be able to be dissolved in something that you can give to a person. That's a drug. And that's what drug companies do really, really well. So Novartis made this into a pill form and sent it to me for testing. And I did what academics do really well. I tested it, and we helped them design and implement the clinical trials. So we each did what we did well, and it all worked out for everybody, including our patients.
But you probably got many drugs, right? So how can you pick up this right one?
So when they sent me drugs, they sent me 5 or 6 compounds. And their favorite compound wasn't the best in my systems at killing CML cells. Because of the work I did, they made it the backup compound to their lead. And their lead compound, they couldn't formulate. And they had no data on this backup compound except mine. And the only data they had for it was in CML. So that became now their lead drug in the lead indication. So there was a little bit of luck in all of that. If their lead compound had gone forward, it would have actually been used broadly because it was a PDGF receptor inhibitor. And that's the activity they really wanted. But the imatinib had better ABL inhibitory activity.
All right. Another questions?
Dr. Druker, thank you very much for your inspiring talk. I have a question about the medicine you use, imatinib, is effective in treating 90% of patients. Then what happened to those less than 10% who do not respond to imatinib? Do you find any genetic difference between those who respond to imatinib and those who do not? Have you now figured out a way to create another medicine to treat specifically for those patients who do not respond to imatinib? Thank you very much.
There's a fair bit of study looking at people who respond less well to imatinib. And what's clear is that most of them have other mutations present at diagnosis. So either they may have a kinase resistant mutation or another pathway that's activated. And so for the kinase mutations, we have lots of drugs for that. For the other pathways activation, we don't have very good drugs for that. But we can at least identify patients that might need to be watched very carefully, would be candidates for a stem cell transplant, which was a prior therapy. So we can actually have some insight into who, again, not surprising, people with multiple molecular abnormalities are less likely to respond to a single agent. So again, not surprising.
Cliff.
I think that we agree that with Minchi that we need to have the people here, the youngsters, need to have a big heart to discover big. But we also know from your story and Andy's story that perseverance is also important. However, just between perseverance and stubbornness is really the neighbors, right? Now, your story talk about the discovery of past 20 years. But nowadays, what this generation facing is a lot of omics, informations, and the speed of discovery actually will be much faster. But also, the solid tumor is much more complicated than liquid tumor. I probably would like to hear what you or maybe any opinion about what do we do if we want to discover something big like Minchi inspires us to do for those people who have all the tools today, and how do we deal with it?
Thank you so much for this great question. I don't think Brian and I have any answer to that. but I can give you some of the historic perspective. Because since year 2011, it seems to us we are now in a new era. It's immunotherapy. We have seen a lot of immune checkpoint inhibitor working on more than 20 different kinds of cancers. So to me, it seems to be a complementary. Because what Brian is saying is that if a cancer with a single driver mutation, then you can identify a compound that inhibit the function of the driver mutation, and you can cure the cancer. But as an example, in Taiwan, one of the most important cancers is liver cancer. And we have been working for 20 years, and we could not identify any driver mutation for liver cancer. So it seems to us there are two kinds of cancer. In one extreme, like CML, you have very clear-cut single driver mutation. And on the other hand, there's another even bigger group of cancer. They do not have any driver mutation. So that's why it seems to be quite natural that immunotherapy come into the scenario. Because even the cancer with driver mutation, they still have a lot of mutation. But in the past, it was called passengers, bystanders, innocent, means they are useless. But now these mutations, although biologically, functionally, they are useless, but they are actually still responding and reactive to host immunity. They are something new, so we call it neoantigen. It's still a mutation that is useful for treatment. So it seems to us we are now in an age that we have molecular target therapy and we have immunotherapy, and these two seem to complement each other. The immunotherapy are dealing with those without any driver mutation, and targeted therapy aiming at those with driver mutations. And now we are going to develop combination of these two, because even any immune cell has signal transductions, and signal transduction will be affected by a lot of tyrosine kinase inhibitors. And we are now only beginning to understand how to use these molecular agents to help immunotherapy. And I must say we have already gone somewhere. We have achieved something, and soon will be released to really advance our level of control of cancers. But what will be beyond that, I don't know, but there will be a lot of possibility. I leave it to Brian.
So I would look at this a couple of ways. Let's start with advanced cancer. So the reality is that for advanced cancer, we need to do a better job of integrating more data. So it's not just the genome, it's what proteins are expressed, how are they modulated, what does the cancer tumor or the tumor microenvironment look like, what's the immune status. And so if we're really going to make inroads against advanced cancer, we have to integrate huge amounts of data, number one. Number two, we need a whole new set of drugs. I'm sitting here, the discoverer of RB. We don't have a drug for RB. We don't have a drug for RAS. We don't have a drug for P53. Really common mutations in cancer, and we have no drugs for them. So that's when I talked about improved drug discovery, it was a testimony to Dr. Chancellor Lee's work. We need more drugs, and people talk about undruggable. Well, kinases were undruggable 20 years ago. And so if RB is undruggable today, that doesn't mean it will always be. The last thing is, the other thing I mentioned was we have to treat early in the course of the disease. When I started my career, we had chemotherapies, mammograms, and PSAs. Now we have all these targeted therapies, immune therapies. For early detection, we have mammograms and PSAs. We can do better than that. We can use this explosion of molecular understanding to identify cancer at its earlier stages where it's less heterogeneous, and our treatments are going to work better. So we have to make investments on both sides in both this advanced cancer with combinations and a better molecular understanding in drug development and discovery, and we have to diagnose and treat cancer earlier.
Ying-Chi, do you want to? Yeah.
Okay, so I think that this opened a lot of interesting issue now. You're talking from liquid cancer to solid tumor. Maybe I can quickly just follow up what Brian just said, that he just mentioned P53. And there are a lot of BCR-ABL is the best by far the example. However, there are a lot of elephants outside there. The two major elephants in the current area, at least I know, P53, Brian just mentioned, RAS oncogene, which has been cloned for identify for 40 years, which don't have effective drug. So there are a lot of opportunity there. So I will also take this opportunity to encourage, especially the youngster here, to think impossible possible. Brian's case already, he's physically here. He's giving a talk. Later on, you're going to come to shake hands with him. He already made impossible possible. That CML, when he gave a talk, he said that before he developed the drug, what happened? If physician tell the patient, say, you're going to live 3 to 5 years, period. But after Gleevec or this drug been developed, now the 5-year survival rate is 90+%. So that's impossible possible. Now, let me come back to solid tumor. And another type of kinase, for example, HER2 new oncotyrosine kinase, that is being known to be overexpressed in about 20% of breast cancer. In a way, very similar to CML, it's about 20% of the entire leukemia. HER2 knew this particular gene at the time in the mid-1980, when the gene was identified, being cloned. I was lucky that I had the opportunity to work with Bob Weinberg, and then at that time involved clone the new oncogene. That at that time, those breast cancer or ovarian cancer patients who have HER2 new has no cure, just like a CML. But nowadays, for breast cancer patients who have HER2 triple positive, overexpression, your 5-year survival rate from hospital to hospital slightly different, but your 5-year survival rate is 60 to 80%. Not 100% yet, but it's very significantly improved. So that's a good news. Another one, which may not be as good news, lung cancer. Lung cancer, regardless male and female, these are our major cancer type. And the lung cancer has a specific mutation, EGFR receptor mutation. And this mutation is even more interesting that because between Asia and Caucasian, the mutation rate is different. That's another big subject. Why in lung cancer, non-small cell lung cancer in Caucasian, about 10 to 15% have EGFR receptor activation mutation. However, in Asia, it's a 50% mutation. And this particular disease has tyrosine kinase inhibitor, similar to Gleevec. However, it's not as easy because lung cancer is a solid tumor, as I just mentioned, and there's a heterogeneity there. So the first generation drug help patients to live 1 year, after that recurrent. Second generation, another year, after that recurrent. Third generation, just being approved 2 years ago, another 1 year, recurrent again. So all the patients who are there are waiting to think, either die or the fourth generation of drug. Now, I'm trying to answer the question. If you want to pick up something very important, there's one project right over there. How can you make those patients, in lung cancer patients, a large number, but mutation, and after third generation of drug, they are recurrent, how can you deal with them? There's an important project there already, and you can think about it, make impossible possible, and follow Brian Druker. 10 years later, you might get the Tang Prize.
Nuhai, you want to make some comment?
In response to Chris' question, I think all those finding good target, finding new drug, precision medicine, all right into do and should do it. But I like to find out a very different mindset. I think we don't need to cure cancer. We don't need to cure every single cancer cell. But we should think also, how can we live with residual disease without clinical symptoms? There's not an answer yet, but this is an area I think we should think more and more, because current therapy cure 99% plus of the cell. How can we deal with the residual? And there are some cancer can be dealt very effectively. Some may not be, like lung cancer may be a tough one. And there are other cancer, one could live with it for life. Think about that.
Thank you. Let me summarize a few words, okay? I think, I'm working on a cancer for my entire life, okay? But I haven't, not like a Brian, I haven't saved any single patient's life. That I feel so bad, okay? You see, that's the reason I cannot get 10 prize. But anyhow, when I sit in this corner, I think from Annie, from Brian, from Mingqi, and from Nuhai, I feel that kind of excitement. Okay, what that means? That means that we have one day able to prolong our lifespan until 150. Because one day we are able to really cure the cancer or live with the cancer in a long time. Then, of course, we can live much, much longer. So that makes me very happy. So because my grandchildren just 1 year old. Okay, so I did have to live at least 40 years so I can see him become, a physician. So that's exactly the story. Okay, I think I want to ask, you have questions? Okay, go ahead.
Also a newcomer in the cancer research field, so I only ask simple, maybe stupid questions. Okay. Many people think that Taiwan is unique because the environment, the humidity, the weather, something is pretty awkward or unique. So how is the experiment made in Taiwan able to represent like universal issues? How is that possible?
You mean geographically, Taiwan is very unique,
yeah.
And so how the Taiwan's case can replicate it in the other part of the country, or how the other part of the country can use in Taiwan? Oh, okay. And let me give this question to Annie. Annie can address that. Okay.
Thank you for these very important and interesting questions. I think in Taiwan, if you are doing very basic work, that's a generalized science. So there's no borders. But you are working on a specific type of cancer. For example, I'm working on liver cancer. And then this is quite endemic cancer. It's not prevalent in Western countries. So this is what you say, if you are working on liver cancer, the cancer here has different etiology. In Taiwan, we have hepatitis B as a major cause of this cancer type. But in Europe, they have alcohol as a major cause of liver cancer. Right. So you have different etiologies to begin with. So probably you would change your angle of research. That's possible. So I must say your question is very important, but if you are basic scientists, I believe most of your work can be generalized to the world. You work on retinoblastogen in Taiwan, that's also good for United States, right? So there's no border. But if you want to work on nasopharyngeal carcinoma, if you want to work on liver cancer, then you have to consider some of the probably geographic difference.
I've got another question that many of us are afraid to go into the lab because we don't have enough background experiences or something. So what age or what stage should we step into the lab?
Okay, this I can answer for you. Okay, this is very important question. The question is, she's very afraid to enter the laboratory, okay, because the laboratory looks like a so mysterious place. It's not like a theater, okay. So when is a good time, good stage to enter the laboratory to learn all those so-called experiments? I hope you did not take a nap during Brian's talk. The first slides Brian showed you, it's middle school. He already got the scientific.
I understand
price.
that the doctor went into the laboratory in high school, but in Taiwan we don't have that much experiences of doing experiments. Okay. So we are afraid of going into the lab.
So I think Brian wanted to help you, and after that I can help you too. So Brian, let him go first.
All right. So despite my picture from middle school, the reality is that I really didn't do serious lab work until I finished all of my medical training. So I'd done a little bit of lab work during my undergraduate, a little bit in medical school, but then I did all of my internal medicine resident training, all of my oncology training, and then I went into a laboratory. So here I am, I'm a board-certified internist, I've finished my oncology training, and I walk into a lab, and I spent the next 8 years in that laboratory. My first year in the laboratory, I was learning about something called acutely transforming retroviruses. I had no idea what those words meant. And I was with postdocs and grad students who were running circles around me while I was trying to run a DNA agarose gel. And I'd go and I'm a competent doctor. I can take care of patients in my sleep, but I can't run a gel. I can't isolate DNA. I don't know what acutely transforming retrovirus means. But I wanted to do this because I wanted my patients to have something better than they had today. Oh, wow. So my point is, there's no wrong time. Any time is okay. You have to have the courage, though, to take that step and think about doing something bigger and better.
I mean, I'm already in the lab of half a year, shadowing some experiments. But some of my friends, my classmates, they are afraid.
I understand that. I think that's the reason someday you will be the 10th prize laureate, but some of your classmates will be another kind. Okay, there's nothing wrong with that. All right. So some may be the CEO of the biotech company that handle the money. Okay, and is try to persuade give the drug for trial. So your classmate may be the CEO of the company, said they know how good you are, they will give you the drug and they do the trial. You see that? There's nothing wrong with it.
All right. Can I follow up this question? This is a very important and interesting question because every person is different. Yeah. You and your classmate are different. And I always feel that I encourage youngsters to think about what area you want to go. Choose from your heart. If you are interested in science, if you are interested in medicine, and because you're interested, you will be allowed to go into the lab. But if you are not interested in experimental work, when you see the mouse, you're scared, but you like a karaoke, and you like the music, you want to go to be an artist, that's fine. So this is probably in another bigger way of thinking about how to choose your career, is to choose something you really, what you like to do, what your heart want to do. Brian, who is a physician, and he has a reason why he chose to be a medical doctor, and he's seen as a leukemia patient. His heart is trying to help the patient, then whatever way to do it. So that's where he comes from. That because you need a drug, which is, at that time is not available, but he sees the patient has no cure, but he want to help them, and from there, he moved toward that direction, he got a success. So it's really depend on what you're interested in. I think it's important, go after your heart.
I think I also want to mention this is a period of time indeed. Brian is very different. Okay, he spent in a very basic laboratory called Tom Roberts. Okay, I know Tom. Okay, working on the polyoma middle T antigens. That's in our field in terms of the DNA tumor virus, RNA tumor virus. That is referred to. Okay, so you understand this kind of basic mechanism from your fundamental understanding. And later, you can think about how you can apply this kind of understanding oncogenesis to the later in your field. Because he's specialized in the hematology oncology, means cancer formations. So that's the reason that he wants to go through this one. Instead of it immediately jump into the clinical practice. That many people do that, but nothing wrong with that. That make Brian is so unique and so distinctive and so to the end, they are all go to the laboratory, go do the research and try to figure out what is the better way to treat the cancer. Today, we have so many medical students sitting here, so many PhD students here, which, like Ming-Chi just said, thinking, listen to your own self. What do you want to be? If you want to make a lot of money, okay, I congratulate you, because CMU doesn't need a lot of money. So later you donate back. Okay, nothing wrong with that. Exactly. But if you think that you really love science, you really like your job to be a physician and take care of good patients, then be prepared, because the current many of the difficult diseases you cannot do anything about. And if you cannot do anything about as a competent physician, you always think through your brain, how can I help patient? Okay, that it's your time spent for. That's your training will be worthwhile. Okay, and otherwise, you go to the laboratory, learning all those things in here, put every time together. When people see in the karaoke, you are in the laboratory, run the agar's chair, okay, it's not worth it. Then you better go to sing a karaoke. Okay, because people think when I run the agar's chair, okay, I want to understand what is going on. I have my dream to carry out. So that's all today's about it. And I hope that many of you are so smart. In Taiwan, the medical student is 1% of these given generations. So as a chancellor, I have really great expectation from them. And so one day, this position sit next to my left-hand side is you. Do you know that? That's you. Okay, I think we had the question from that side, okay? A lady. Okay.
While you're waiting, I was going to add one more element to success. And my colleagues have talked a lot about passion and following your heart. I'd also add talent to that. I mean, it helps to have a little bit of talent in what you're doing. And I'll just give you my example. And Sherry will tell you that she thinks I'm tall enough to be a basketball player. And I love sports. I love basketball. I'm terrible at it. And no matter how hard I work, no matter how many hours I put in, I will never become a good basketball player. But I could be passionate about it. So if you really want to go far, you need to combine talent and passion, and then the sky is the limit.
Good saying. And then, oh.
我想请问一下啊，大家是从小开始研究了那个案例小女生目前几岁了？然后，人要从小教啊，商要从小栽。那我想请问一下，说，酪氨酸激酶可以抑制癌症基因，这个小女孩如果一直长大，会继续的追踪她到生孩子吗？啊，未来会不会说利用中草药，去抑制，用了酪氨酸来抑制癌症基因？那可不可以也有，人体自己免疫性的研究，配合女性的荷尔蒙，比如说有的人男性荷尔蒙也会互动。如果我们像月经这样子互动的话，可以用个体的一起符合你的那个医药一起继续研发新的案例吗？
Okay, I will translate your questions into English to Brian. So I think her first question is, how old the young lady that when you treated when she was 6 years old? How old she is now?
That was 2001, so she's 23 now.
23 now. Okay,
可以
and
帮我
then
翻译一下吗？
我再问第二个 question. Okay, and then I think that she asked the second question is, if how long you expect, and if she is pregnant, and how can the deal to take care of this one? Yeah.
We know I've taken somewhere around 25 or 30 women through pregnancy, with a total of around 50 babies being born, because of our concerns about the drug causing fetal abnormalities. As soon as somebody becomes pregnant, we'd stop their therapy and observe them during their pregnancy. And because we know that by the time they get to the third trimester, the drug would be safe, if they need to be started back on medicine in the third trimester, we've done that. And so far in my practice, I've not seen any birth defects. In the literature, there may be a slight increase, but I think the reason for that is that the women are exposed very early in pregnancy to the drug, whereas I've been very careful as soon as pregnancy is detected to stop the medication and at least get to the third trimester. And I've not either I've not seen any fetal abnormalities, and we've not had any patients lose control of their leukemia when they go back on their medication.
Okay, and I think, of course, here she also asked the questions regarding about Chinese medicine, but you don't know how to answer. Could you? You know how to start. And then you answer. You say, can we use Chinese medicine to, replace all this grieving?
Yeah, I don't know about all of Chinese medicine, but one of my very favorite stories was about the use of arsenic for the treatment.
No, now you can't. Yeah, that's very toxic. Go ahead.
Yes, but the story of arsenic for a particular type of leukemia was pioneered in China. And at a very famous conference, the investigator was asked, how do you know what dose to use? His answer was 2,000 years of experience.
这个小女生已经23岁了，好，第一个问题。第二个问题呢，就是这个，假如你这些得病的这些人呢，假如他一开始怀孕，你就把药拿掉 ，OK， 一直等到他 trimester 叫什么？应当怎么办？到妊娠第3期的时候再处理就可以了，这样子。那现在他照顾的50几个这些妇女呢，都没有生出来孩子都没有多大问题，但有一小部分，假如你早期怀孕呢没有处理好，它是会有问题的。哦，第三个问题就是有关于那个中草药。中草药最出名的治血癌最出名的就是砒霜 ，OK， 砒霜 ，OK， 所以这个东西呢很毒。那要用多少量呢？那2000年的历史呢？因为这是中国人的药嘛，对不对哈？所以已经是知道要用多少了。吃多了就会死翘翘，不要忘掉了哈。只能吃一点点。这 这个我想医生们都知道，可以了哈。好 ，OK， 来 ，we have the OK now we inspire all the students want to ask question now. OK, go ahead.
老师好，因为我不会讲英文，所以我是用中文发问。我的第一个问题就是因为伊马替尼是透过抑制酪氨酸激酶来抑制癌症，那所有的癌症都有酪氨酸激酶的异常情况，那应该伊马替尼可以杀掉所有的癌细胞才对，但现实中好像不是这样，为什么呢？然后第二个问题就是说，因为现在有一些新研发出来已经上市的药，就是可以抑制癌症的其他讯息的传递。那酪氨酸激酶这条途径还有其他的替代途径，所以癌症会产生抗药性。那如果说把所有关于这方讯息传递路径的药都结合起来，变成鸡尾酒疗法，这个可以吗？第三个问题是，因为酪氨酸激酶是负责传递生长讯号，那干细胞、癌细胞都会用到这个机制，伊马替尼应该会对干细胞产生影响，但好像没有发生这样子的现象。以上是我的问题，谢谢。
Ming-Chi, you want to try? Let Ming-Chi to answer. Okay.
So I think the first question you probably already know the answer. 你要用中文回答他。
对，用中文。
你第一个问题说酪氨酸激酶，对不对哈？因为这个Gleevec 是一个酪氨酸激酶，那么酪氨酸在很多癌细胞都有，对不对？为什么这个Brian develop 这个药不会把所有的 cancer cell 都消失，只能治疗这个 CML？ 你是不是这个意思？对不对？好，酪氨酸激酶只是我们这样讲，它是但是酪氨酸有很多那个酵素有很多不一样的酵素。它现在这个药呢，是它对这一个 BCR-ABL 的这个酪氨酸激酶对这个的抑制的能力呢，比对其他的酪氨酸激酶还强很多。所以这个药主要是 target 这一种癌细胞。那么另外一个癌细胞，如果它是另外一个酪氨酸激酶的话，但是它不是 BCR-ABL， 那么如果你要用这个药去杀死另外癌细胞，你这个药的浓度可能要高很多，可能要高到50倍、100倍。可是，在治疗病人的时候，我们不是这样治疗。如果说另外一个酪氨酸激酶，你就要再去发展另外一个药物，它会有一个所谓叫做 specificity， 就专一性。所以这个药是因为对 BCR-ABL 这个酪氨酸有专一性，那这个 BCR-ABL 正好是 CML 这个病引起这个病。如果是另外一个病的话，那就要另外一种不一样的化合物，但是也有专一性，就可以治疗其他的病。这个是第一个问题。What's the second question？ 第二个问题，你说是是 signal pathway 是吧？对不对？对，你这个观念可能是对的。就是如果我们不要用那个 leukemia， 因为 leukemia 是 liquid cancer 比较 simple， 像 CML 就是一个 BCR-ABL。如果你用 solid tumor， 用那个肿瘤，比如说用乳腺癌啦，或者用肺癌啦，或者其他的这个 solid tumor， 因为它的那个 tumor 的一样的一个 tumor， 不同的 tumor cell， 它可能有不一样的 mutation。比如说假如一样的乳腺癌，那这个病人这个乳腺癌呢，它100万个 tumor cell 里面，可能其中有一些是 HER2 mutation， 其中另外有一些 tumor cell 可能是 c-MET 的 mutation。那在这种情形之下，就像你刚刚讲的，因为这些 signal 这些细胞传递的讯息，是不是可以做 combination？ 假如将来我们很好的方法去 detect， 现在可能已经有了，就是 solid tumor 里面不一样的 tumor cell 的突变的话，那么在那种 solid tumor， 你是可以用联合治疗来做治疗，是可以的呀。
第三个是 stem cell。
第三个问题
是干细胞，对不对？
第三个问题就是说，酪氨酸激酶它是负责传导生长讯息的。那癌细胞因为酪氨酸生长讯息的 那个控制失控，所以就变成癌细胞。但是干细胞它要不断的分裂，还是必须要用到这个过程啊？为什么伊马替尼不会对干细胞造成影响？
你现在的问题是说，酪氨酸激酶会抑制癌细胞，同时也会抑制干细胞，是吗？好，那你是说，当你用这个酪氨酸激酶的抑制剂在抑制癌细胞的时候，是不是也会抑制干细胞，是吗？对，是有可能，但是记得专一性就是这个药，这个病人就用 CML 的病人来讲，这个 BCR-ABL， 因为它是这个 BCR 这个酪氨酸变突变了，所以你现在用的药是专一性，只对这个。那么正常的干细胞呢，它可能 BCR-ABL 没有 mutation， 它不一定会这个对这个专一性的这个药有会被抑制。就像我刚刚跟你讲的一样的观念，不一样的癌细胞，对不对？你以为说啊，那我用这个药可以杀死 CML， 我也可以杀死其他的，因为其他的癌细胞它没有 CML。关键就是我们现在所谓的标靶治疗，就是说你 design 一个药对症下药，就这个药专门对这个癌细胞的那一个酪氨酸，但是其他的细胞、其他的癌细胞或者其他的正常细胞，它这个酪氨酸如果没有突变的话，是不会受影响的。这样有没有回答你问题？好，谢谢。
那这样等一下，我刚刚把你的问题呢用英文translate，把它用英文呢翻译成给 Brian 听，Brian 听完了以后呢，他要回答你的问题，我再转译给你，好不好？Brian, please go ahead. As I just said that, I will translate what you said.
Yes. Okay. If we could figure out why imatinib doesn't kill the CML stem cell, we could eradicate this disease. Why don't you come and work in my lab and figure that out?
他说 ，OK， 这个问题很好 ，OK， 那么要你呢到他实验室开始做这个题目，把它做出来，说为什么这个药呢，它不会把这个干细胞把它杀掉，好不好？好不好？你看大师要 offer 你一个工作 ，OK， 妈妈在哪里啊？妈妈怎么带他去 ？OK。在这里哈，好。刚才我们还有2位同学，来，同学们 ，OK。
Hello, I'm Jack, and I'm a 5th-year medical school student and a physician to be in 2 years. And so I have 2 quick questions. Like first is about environment. So I knew that you do your MS in San Diego and the clinical training in Washington U and a fellow in Harvard, but why eventually you choose Oregon, OHSU, as the place that you do your research? So the first part is the environment question, and second part is the leadership question. So I know that this is a really big project, and to do the target identification, you must have a really good team. So what are the criteria for you to choose your team member? And what are those criteria? And I know he can join the team. So I knew that courage is one big thing, and also young. Okay, thanks.
So the first question is, how do you choose where to go and work? And for me, it was, I wanted to find a place that was committed to cancer research, was growing a cancer program, and I wanted to be part of something that was growing as opposed to something where it was much larger and it was harder to find your place. So I found, and the other thing that I learned was you need a really good boss. And I had a wonderful mentor who established the cancer program in Oregon by the name of Grover Bagby. And he was a person who, at the time when Harvard was saying I didn't have a future, he looked at me and said, I think you do. So that was a good environment. It was an up-and-coming program committed to cancer research. It was a place where I thought if I made a difference, it would matter. And I had a wonderful boss. So all those things made Oregon a great choice for me. And the second question was about leadership. I've just been so incredibly lucky to have great people working in my laboratory, but people that get along really well with one another and support one another. And I wish there was something I could say that I did to make that happen, but I've just been really lucky to get great people wanting to work as a team and get along really well. And that's been, when people come and look at my laboratory and they say, we just love working with your group, that makes me really happy and proud. I'm as proud of that as I am of our accomplishments, because if people enjoy their work, enjoy their colleagues, they're going to be more productive. And again, I don't know that I've done what I've done to deserve that, but it certainly is something that you want to try to aspire to as a leader, is to surround yourself with great people, but also really good people.
Did that answer you? Yeah. Okay. I think you will be a good team member. Please join Brown's laboratory. Okay. And that's the key. I think that, every student in this university are goddamn good. Okay. They just need a good environment to cultivate. So when you put you in Harvard, you will be as good as any student in Harvard. That I believe so. Okay. And that's, you have to this confidence. You have to have this confidence. Okay, so then you can see how they behave. I think I'm quite sure you will behave as good as they are. Now, we just sent 2 students to the UCI. Okay, and then 2 professors say, God, this is so good students. So why you only send 2? How about you send 20? Okay, that is the true story. Okay, so be confident about that. And that's, I'm looking forward, okay, for all of the best students that you are excited about your future, and you should go to pursue it, okay, your future to pursue it. Okay, there's another question for the next, please. Okay.
Thank you, Doctor. I'm a 5th-year medical student, and I would like to ask, what is the most challenging or the most difficult thing to you in your academic career? I'm very curious about that. Thank you.
Well, I think I've covered at least one of them when I was leaving Harvard. But the other really was, again, something we've covered, which was trying to convince a very large drug company to go to clinical trials. So I got the compound in my lab in 1993. By February of 1994, I had pretty much completed the testing. It took me 2 years to get it published because it got rejected at journals like Science and Nature, and they thought it wasn't reproducible and too clinical. But so I had this data in February 1994. We didn't start clinical trials until June of 1998. So for 4.5 years, I was trying to convince a company to move this out of the lab into the clinic. And those were really frustrating years, because during that time I had patients in my clinic that died of this leukemia. And if we had been 2 years earlier, some of those people would still be here today. If we'd been 2 years later, some of the people that I showed in my pictures wouldn't be here. So timing is everything, but that was an incredibly frustrating period of time in my life where I knew we were onto something that needed to be tested, but I couldn't convince a company to move more quickly. But ultimately, having the connection to my patients is what helped me persevere, because they needed something and they needed somebody to be their champion.
That is a really nice say. You need a good friend to be the company CEO. Okay, so when they sing a lot of karaoke, I say, go ahead. Okay, so because sooner or later they become a CEO of the company and they believe you. Okay, so Brian, that was short in the time.
Yeah,
correct.
Great.
Any more? Again, I think that's answered a lot of good questions. Okay, now, come here. Okay, come here.
I'm a dentist, and I'm now a PhD student. I work on lab. After I graduate 20 years. And why I come to the lab? Because I want to resolve some problem I cannot resolve in the clinic. when we talk about tumor, we know that the carcinogen is most in the environment of our life. But when we talk about gene, the possibility may be 1, 2, even to 10. Why we do the best effort to do the our effort in the maybe gene is the maybe 10th of the carcinogen. Why do we make the effort in the environment to prevention? My father got cancer, urethra cancer. And when we talk about how to treat, because that's a lucky, because he got allergy. So he take many medicine and cannot to resolve the itching all of the body. But after examination, noticed that urethra had the cancer over the urethra. But the doctor said, you must to resolve this problem, you must to cut off the urethra and the kidney, because we have another material to connect the urethra. But many doctors say that even we go to the Taida, maybe the best in Taiwan. But and many physicians say that you can take away one kidney because our metabolism only 1/4 of the kidney, 1/4 is enough for our metabolism. But we think if we only want to have the 1/4 of the kidney, why God give us 2? Why? We don't know. The best doctor is God. So we pray. In our arch, we pray. And we found one physician in our downtown. His research is to reconnect the urethra. So he only cut off the urethra and reconnect. And after that, my father itching is gone. We use acupuncture, and my father is better than before. And now he quit smoking. Before he quit smoking for 100 times, but now he's really quit. So I mean that knowledge is good, but if we must to cooperate with another and keep in mind the truth. We know that truth is part of the truth. Truth is in our mind. So when our education is about how to treat the result, why we cannot to educate to prevention for our life? Thank you.
谢谢 Thank you. I think you want to answer this one in terms of prevention or the treatment in that way?
So let me talk about prevention, and it's, as you saw in one of my very final slides, I talked a lot about a broad-based approach, that it's not just about specific treatments, it's also about prevention and early detection. And as I think about the future, I also think about what I would call molecularly targeted prevention, meaning understanding our own individual risks, doing a better job of understanding environmental risks. Clearly, there are risks we know about. And you mentioned your father was a smoker. That's a risk factor for many cancers. We know that. We need to decrease smoking rates aggressively. We know that air pollution is likely to become a bigger problem and a cause for cancers. We need to talk about one of my fellow laureates, Jim Hansen, about decreasing our carbon emissions. So there are many things that we need to do, but ultimately, I do agree that prevention is a critical aspect of how we manage cancer and make it more curable, more treatable, and more preventable.
Okay, to answer you. Okay, I think that we take last questions. Okay, that the gentleman raised their hand on the other side.
Since the professor's speech, I have a very short question. I'm a PhD student, but also a medical oncologist. I'm very impressed about your work doing the cancer research and also taking patients. But in Taiwan, the system is a little bit different. So we take patients all day. So we have little time to do our cancer research. So what's your suggestion about my future or our Taiwan's oncology future? Thank you.
He spends most of the time to taking care of the patients, and so there's no time to do that.
Yes, so it's not that different in the United States, where many of our clinicians spend most of their time taking care of patients and often don't have enough time to do research or clinical research. What we're trying to do is we're trying to get more physicians involved in research, offer them more protected time, because when I went through my training, I could disappear into the lab for 8 years and nobody cared. As long as I was bringing in my own grant funding and didn't cost the institution any money, I was fine. But now things have changed, and oftentimes it's more difficult to find that protected time. And at least in the United States, we're coming back to the recognition that if we don't put funding into promising young physician scientists, we'll miss out on a whole generation. So we've recognized the need to protect from clinical time for people to do research. But I'd also one of the other things I'm learning as I'm directing my own center is we have to value people for what they do. So I have really busy clinical faculty, and they're wonderful. And I need to make sure that they feel valued too. And so if I start using words like, oh, we've got to protect you from the clinic, then I'm starting to say, well, the clinician's time is less valuable than the researcher's time. So you actually have to come up with a different way of talking about this, because the clinical faculty are equally as important as the research faculty. Each have to be valued for what they do. Each have to be supported for what they do.
I think you probably don't feel good for this answer, correct? Yeah.
No, it's a very hard problem.
It is a very, very difficult problem. The most difficult part is every physician, after they go through this long years of training, they expect their high salary. Correct? Okay, they expect twice, 3 times, okay, this amount of salary. And so where the salary come from, okay, see the patients. In the US, you supplement by grant. Okay, so therefore you can see less patients because you have the grant to pay you at time so you can do a research. And Taiwan, we don't have such mechanism. And so we have to work one side to take care of the patients. The other side, do the research is for fun. Okay, for fun. But there's no fun. Correct? You don't have time to do it. And that is our problem. okay, our current problem. And I'm fully aware of this one. So that's the reason each time in the minister meeting, I always deem them. Okay, we have to protect, okay, the young physician scientist time. We say that it does not mean that clinical time is not important. It's we need to protect in order to reach the equal footing in terms of payment. So for example, everybody get a 30万 or 25万 25万怎么讲25万 okay, 250,000 NT, for example, a month. Everybody get equal. Okay, but you do clinical work, and I do part of clinical work, and I also have time to do a research. And so this among the research, and we can come from the Minister of the Technology and Science, most, okay, to supplement you, supplement university, so you can do a research. Can we do that? Okay, let us work on that. Okay, from the 卫福部, okay, also from the Koji部, that we have a chance to get this done. Because that is the future for our young generation to go into the research, called as physician scientist. That I'm very, very keen about this part. So that's, we have so many important people here, so we all speak out, we have a chance to get it. Right? I mean, yeah.
Well, no, can I just add one more point? I think that you listen to the answer. This is an important issue that everybody should try do their own job. And therefore, I thought there's another very important factor. That is, if let's use cancer as an example. If that's your goal, that it is important to learn how to work together as a team. Because it's virtually impossible, one person can do everything. So every person has their own specific function, pure clinician, physician scientist, or just laboratory science. And people are in academic, in industry, and in government. So for a disease or for any major issue, teamwork is important. And while doing our training, we all had to learn how to work with other people. And there are a lot of skills for that. For example, one of the very important ones is respect to each other. Keep in mind, there's a Chinese saying, 文人相亲, scholars always look down upon each other. Who's the best? I am. The other person, piece of shit. Okay? Don't have that kind of attitude. Respect each other. And therefore, you have a common goal, then you work together to get things done and share appropriate credit together. That's my personal opinion.
Yeah, that is a very, very good advice. I think that all the students listen to that. It's very important to have the team and respect each other. I think he's mentioned about too much work. I aware of that. But okay, you have to prioritize what you want. Okay, so then you can get the things done. Okay, I think we have enough time for discussion now. So I think we probably have to. Right. Okay.
Now we would like to invite...
Close the discussion. Thank you very much.
Thank you. And now we would like to invite Chancellor Lee to give us a closing remark. Right. Yeah.
I think, first, I really thank Tang Foundation, okay, give us this opportunity, okay, to host this important event. I hope through this event that we have Brian here, we have Andy here, we have Mingqi here, Luhai here, and give us a lot of the so-called experience and the lesson. And through that, our students can learn it. And through that, our students can have the confidence that you can be as good as anyone. So as soon as you are decided and pursued with your own talent. Okay, that's really my hope. Of course, I think I can see how enjoyed that this group of the audience in terms of discussion and in terms of raising all the difficulty and all that stuff. There is indeed a lot of work, okay, a lot of obstacle in front of us, but nothing comes with easy, nothing, okay, nothing comes with easy. As soon as we work together, okay, we will overcome any obstacle and to achieve your goal. Thank you.
Thank you, Chancellor Lee, for hosting a wonderful session. And thank you all for your active participation. Before we stop, we have two things to do. The first, we would like to invite Chancellor Lee to give appreciation souvenir to our speakers and all panelists. And we would like to invite CEO Zheng Chuan of the Tang Prize Foundation to the stage to receive the appreciation souvenir as well. So this is a souvenir from Tang Prize Foundation to CMU.
On behalf of Tang Prize Foundation, I'd like to thank the President and the CMU for fostering such a wonderful forums. But also, I remind you that we have, besides wonderful Brian Druker, we also have several other outstanding laureates. This year, we also have John Madison and Tony Hunter. Okay, 3 lawyers received the joint award for this year's Tang Prize for Biopharmaceutical Science Award. So please go to our web and look at their stories. That can inspire you very much. Yeah, thank you.
I think I plan to give this so-called souvenir Gao Liang Jiu. Okay, that's all our 60th anniversary. Okay, for the university, and so this is an item you can collect. 60 years later, this price will be enormous. Okay, right.
So thank you, Dr. Druker, for your spectacular talk and sharing with us. Thank you. Thank you, Superintendent An Li-zheng of National Taiwan University Cancer Center, for your discussion. Next, we would like to express our appreciation to Vice President Ming-Chi Hong of the University of Texas MD Anderson Cancer Center. Now we are going to the second thing, is to have group photo. Oh, not yet. Here we would like to express our appreciation to Vice President Lu Hai Wang of China Medical University.